Oh. Howdy. If you're watching this tape, then congratulations. Your application for Red Ribbon Robotics and Biotechnology has been reviewed and accepted. My name is Axis, the star of this fine establishment. This video will be showing you what you will be doing as the architect manager and how each component of your facility works. But enough talk about introductions. Let's get down to business, shall we? Let's start off with something a bit obvious. The exterior of your new home. You've manufactured these walls to be made out of an alloy of steel, iron, nickel, or as I like to call it, sin. I like to think of these walls like the membrane of a cell, as they are the main walls that keep everything contained and protected, also allowing certain passengers who want to come by the facility to enter or exit depending on what they are. Moving into the first floor of our lovely facility, we have our tremendously powerful core generator. This generator here is what gives power to the entire building, so any small mess up could lead to the building going dark. Dark humor aside, try to think of its core as our mitochondria, as we use this lovely piece of work as our main power source. Next up on our list will be the numerous as highly advanced citric acid sprinklers. For the uneducated, at Red Ribbon Robotics, we use citric acid for the specific reason that it will only get rid of the rust and other unwanted substances that grow inside of our lab. It's a super fast and efficient process, as we expel all the unwanted attachments in return for a cleaner future. Now, for some of you, these sprinklers might remind you of the lysosomes that are found inside cells, as they exist for the exact same reason, to clean the interior of the cells and get rid of the excess waste. Ow. Okay. Well, that hurt. Adding on to our sprinklers though, another big thing that was added to our establishment were these drainage holes that are designed to drain any and all liquids that are spilled in our laboratory, including the acid that helps clean this place up. Think of these sinkholes as our vesicles, both of them having the same function, but unlike lysosomes whose function is to only clean the mess, vesicles are the ones who actually take the waste and throw it outside of the cell. We do advise to use caution in our lab, as stepping into the wrong place at the wrong time could lead to some unintended results. But my point still stands, kid. Don't make any dumb decisions. Moving away from how our cleaning processes work, we have this lovely little container that houses these adorable little critters. Sorry about that. They're not really all that used to strangers, are they? You're probably wondering what these creatures even are. Well, these are called Yochu, or Kitaru Saibo, if you're going for their scientific name. When they're in a humid, cramped, dark area like this, they like to secrete this gel-like solution via their mouth. Now, I know that might sound disgusting at first, and further research is still needed to know why they do this, but it's actually a really good source of energy. With a bit of testing, we found out that this gel is actually made of pure proteins, which is one of the main ways that our facility is powered. Now, if you're a science whiz like I am, you probably noticed that the Yochu are like living ribosomes, producing proteins that the cell or facility can use. Now, you might be wondering how we are able to take up these proteins and turn them into pure energy. Well, my friend, that takes a few more steps than you might think. This next machine is what I like to call the Nectar Capsule Packager, or NCP. Its function is pretty simple. The nectar gets transported into the conveyor belt, taken as part of a sleek capsule, and gets moved into the tubes over there that are connected to the core. I'll get back to that later though. Think of this conveyor as our Golgi body, packaging the proteins that are made so that it can be sent off to our core. Now, remember those parts that I mentioned? Well, we will be discussing that next, and this is the last main mechanism for the protein's journey to give us our power. So you may be asking yourself, why are there so many different pipes that go in different directions? Well, I can explain that in one word: efficiency. Think of it like this. If we had designed it so that it was just this one-way straight shot to the core, there would be only one way for the proteins to reach the core. Plus, if too many proteins are 1 and 2, it raises the likelihood of that pipe getting clogged. And by making multiple paths for the capsules to go down, we are making more of the capsules reach the core without any issue. Kind of like the rough endoplasmic reticulum of the cell, giving us a large pathway that the capsules can get to through the core. The last thing I will mention to you before we need to go on to our second floor is these illustrious tungsten pillars. So, anyone else hear that real thing?
ごめん、あまない。
When we were first designing this laboratory, we realized that the top floor actually had a big tendency to break and lose stability over on the north side, and we chalked up the cause to being the main core, as all the circuitry putting too much weight on the floors. Now, redesigning a new core was the last thing any of us wanted to do, so we had the idea of adding pillars to our building, as this was the only way to stay stable. Cytoplasm acts very similar to these pillars, as their only job is to give stability to the cell and to make sure that everything stays in place. Well, now that we have finished discussing what is on the first floor, let's go ahead and move on to the second, shall we? Let me just... And voila! Here we are, the second floor. Isn't she a beauty? Let's start off with the main elephant in the room, the security system. Now, even though this looks like a supervillain surveillance system, it's not. This one station is able to control nearly everything the lab can do, from the amount of power the core is able to generate to locking unwanted guests out of our building if they aren't in our database. What's with that face? I told you this was our main security system. Try to think of this control center as their nucleus, or the main brain of this building. Moving away from the main thinker of our lab, we have this lovely little box that I like to call our hatchery. If we take a look inside here, we can actually see that this glass chamber is filled with these small bubbles that want to grow, but they are unable to due to the solution they're being kept in at the moment. These are actually the cells from the Yochu, and this is truly fascinating, as these creatures actually reproduce asexually, and there's surprisingly no genetic alteration every time one of these matures. Now, once in a while, one of the cells is dropped into this bottom chamber, where it is shocked with a small amount of electricity. This jolt forces the cell to start the process of meiosis, and in a matter of a few hours, a new Yochu egg is born and can be dropped into the chamber below at the bottom floor, and the process starts again. This hatchery is like a nucleolus, creating ribosomes that will eventually make the protein necessary for its survival. Now, for those of you who have a good eye, you may have noticed these hiding things, wondering what they were for. Well, as our main organic manager, you have to be prepared for any and all situations, which includes if something breaks or if anything doesn't work properly. In my time being here, a lot of stuff has changed in our facility, but there's also been a lot of stuff that's been broken. Let's think of these beams as essential for the cell. As many changes happen in a building, these will be the quote-unquote building blocks that will be used to fix any physical problems. I'm sure we can both agree that this tour has been going on for a long time. And believe me, I'm getting pretty tired here as well, so let's wrap this up. Our final mechanism that I will introduce to you is a special storage cabinet that stores everything needed for the hatchery to make sure it keeps running. From the reproductive cells of the Yochu to the batteries needed to control the voltage, you will have everything you need to continue the hatchery. Now you might be wondering how you are supposed to get these cells needed for the experiment, and the answer is quite simple. Anytime one of the Yochu is finished vomiting out their proteins needed to fuel the core, it'll shed clusters of its skin off until it has a new set of skin. Using the control panel above on its floor, it sends the old skin up to the hatchery. Just shove the skin into the slot near the console, and voila! You'll have your skin cells! The best way to describe this storage closet is like the vacuole of the cell, storing waste products that are found inside of it. Well, there you have it, kid. Every major mechanism that the Erector Robotics and Biotechnology Lab has to offer. Seems complicated, doesn't it? Well, don't worry. The job will get easier as you keep trying your best and improving your skills. As our new architect manager, you'll be doing maintenance checks every hour, rechanging the Yochu cells, and making sure that they have a stable amount of power flowing through the facility. I know all this might seem overwhelming to you, but I believe that eventually you'll be able to handle it. I'll be rooting for you, kid. Sayonara!